Today we will talk about cells. Before starting, let's know the difference between these two terms, stalactite and snowtites. Stalactite is a long, thin spike of limestone hanging down from the roof of a cave, while snowtite, it is a long, thin drip of bacteria-filled slime. So, the bacteria which can live in these harsh conditions, they are called as extremophiles. Extremophiles are the lovers of extreme conditions. They are the organisms that can live in an environment so strange that almost nothing else can live there. And the name snowtite that was given by caver Jim Pisarovich. And in the atmosphere of these caves, a smelly toxic gas called hydrogen sulfide is found, which the bacteria use to make their food. In the middle of the threads, there is virtually no oxygen. We cannot find any oxygen there. And these kind of extremophile bacteria are believed to have evolved on Earth around about 3.5 billion years ago. So, coming back to the cell structures, all the organisms they are made of cells. The cells are very small, so large organisms they contain millions of cells. Some organisms are unicellular while some are multicellular. You can see here unicellular organisms, paramecium, amoeba, bacteria, and yeast, and multicellular organisms like human beings, plants, other animals. You can see the hierarchy that the cells combine to form the tissues combine to form organ, and then organ systems, and the organ systems combine to form an organism. Now the question arises, how can we see the cells? Because they are very small. So we need to use a microscope to see the cells clearly. So the light microscope that we use normally, that shines light through the piece of animal or plants we are looking at. It magnifies up to 1500 times. And a photomicrograph, that is a picture made using a light microscope. I mean, any picture, anything that you see using a light microscope, if you take a picture of that, that is called as photomicrograph. So sometimes we can also see some objects with the help of hand lens. A hand lens magnifies 10 times, but with the help of hand lens, if we see the cells, they can often be seen as dots. So this is how a light microscope, which is also called as optical microscope, it looks like. Now the light microscope, it can be classified as simple and compound microscope. In the simple type, single lens like magnifying glass are only used. Whereas in compound type, several lenses are used to magnify the objects clearly. Here you can see in the this is the mirror where the light reflects. It passes through the objective lens, and then we can see here with the ocular lens, with the help of the eyepiece, we can see the object. An electron microscope. Now, the things which we cannot see with the help of light microscope, like the cell organelles, for that we need electron microscope. An electron microscope magnifies up to 10 million times, and it uses a beam of electrons instead of light. And the photograph of cells taken using an electron microscope, they are called as electron micrographs. Electron microscopes only show images in black and white. Here you can see the difference between the images taken with the help of light microscope and under electron microscope. We can clearly see the difference. In the light microscope, we cannot see any organelle, but under electron microscope, we can see the organelles like the Golgi apparatus here, the nucleus and the mitochondria. Now, the features which are common to both plant and animal cells. There are features which are common, like you can see the cell membrane present in both nucleus, then the nucleolus, smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, vacuole. In the plant cells, the vacuole is very large. Mitochondria is there in both. Then in the animal cells, centrioles, lysosomes, and centrosomes are there. While in the case of plant cells, we have the chloroplast, cell wall, and the plasmodesmata. So, from this figure, we can compare the plant and animal cells. Now, cell wall in plants, it is made up of cellulose. Cell membrane, cell membrane is made of proteins and fats. Cell membrane, wherever we find in animal cells or wherever in the plant cells also, it is always made up of proteins and fats. Cell membrane is partially permeable, means that it will let some substances through but not others. It controls what goes in and out of the cell. Now, in the plant cells, nucleus is difficult to be seen as it is right against the cell wall due to the presence of large central vacuole. So we cannot see it very easily. Coming to the cytoplasm. Now, inner to the cell membrane is present a jelly-like substance which is called as the cytoplasm. It contains a lot of water and it contains many organelles, and it is the site of metabolic reactions. Whatever the reactions or the metabolism going on in a cell, it takes place in the cytoplasm. Now here is the structure of the cell wall of plants. As we discussed, that plant cell wall, it is made up of cellulose. Now the cellulose form fibers which crisscross over one another to form a very strong covering to the cell. And this crisscross linking of the cellulose fibers protect and support the cell. If the cell absorbs a lot of water and swells, the cell wall stops it from bursting. You can see here the cell wall, it is made up of cellulose. Cellulose is a polysaccharide. So here, if we start from here, the glucose monomers, glucose combine to form cellulose chains, then combine to form microfibrils and fibrils, and then cellulose fibrils in a plant cell. In the figure, you can see the crisscross linking of the cellulose, and there are spaces between cellulose fibers, so even very large molecules are able to go through the cellulose cell wall. So it is therefore said to be fully permeable. We need to remember the difference that cell membrane is partially permeable where the cell wall is fully permeable. So cellulose in plant cell wall forms fibers which crisscross over one another. Cell wall is fully permeable, and the central vacuole in the plants is surrounded by membrane which is called as tonoplast. And the cell sap, I mean the liquid content of the central vacuole, that is called as cell sap, it serves as a storage of materials and provides mechanical support. It also plays a vital role in osmosis. A plant cell stays in shape with the help of vacuole and cell wall. So these two things, they make the plant cell very turgid. It helps to the plant cell to maintain its shape. So that is it for today, and tomorrow we'll continue with the further topics. Thank you.